In this video, we will be using goldfish and fishermen to explain mitosis and meiosis. Each goldfish represents one chromosome. Crushed up goldfish represent chromosomes that are not condensed. 2 goldfish represent sister chromatids. 4 goldfish represent homologous pairs. The fishing strings represent the microtubules of the mitotic spindle. The fishing net represents the nucleus. The fishermen represent the centrosomes. The cell cycle consists of interphase mitosis and cytokinesis. Cells are in interphase for 90% of the cell cycle. Interphase is composed of 3 subphases, G1, S, and G2, and is where cells grow and undergo synthesis. After interphase, cells undergo mitosis where the nucleus is divided and the chromosomes are given to the daughter nuclei. After mitosis, the cytoplasm divides, producing 2 daughter cells. This is called cytokinesis. Mitosis is important for several reasons. It allows the cells of a developing embryo and single-celled organisms to reproduce. It also allows somatic cells to reproduce. This is important because somatic cells are ordinary body cells and make up skin, hair, and muscle. Before mitosis, during interphase, the cell contains 2 centrosomes and a nucleus. The nucleus contains nucleoli and chromosomes and is bound by a nuclear envelope. The first phase of mitosis is prophase. During this phase, the centrosomes move away from each other and the mitotic spindle begins to form. The chromatin fibers condense into chromosomes and each duplicated chromosome appears as 2 sister chromatids, which are identical and are joined at their centromeres. As well, the nucleoli disappear. The next phase is prometaphase. During this phase, the nuclear envelope breaks down. The chromosomes condense even more and they form kinetochores. Kinetochore microtubules from the mitotic spindle attach to the kinetochores and the non-kinetochore microtubules attach to the other non-kinetochore microtubules from the opposite side of the spindle. Next, the cell goes through metaphase, which is the longest phase. By now, the centrosomes are located at opposite ends of the cell. As well, the chromosomes line up along the metaphase plate, which is equidistant between the 2 poles of the spindle. Each chromosome is attached to microtubules of the mitotic spindle by their kinetochores. The next phase is anaphase, the shortest stage. During this stage, sister chromatids are separated, and each chromatid becomes a chromosome. The kinetochore microtubules shorten, and the daughter chromosomes move toward their respective end of the cell. The non-kinetochore microtubules lengthen and elongate the cell. The last phase is telophase, during which 2 daughter nuclei form. The nucleoli reappear, the nuclear envelope forms, and the chromosomes become less condensed. At the end of mitosis, the cell undergoes cytokinesis, during which its cytoplasm divides. The 2 resulting daughter cells are identical to the parent cell. They are diploid and have the same number of chromosomes as the parent cell.
Meiosis is important because it is responsible for the production of gametes. During meiosis I, homologous pairs are separated, and during meiosis II, sister chromatids are separated. During prophase I, the spindle forms and the nuclear envelope breaks down. Chromosomes condense and homologs pair up. While homologs are in synapsis, crossing over occurs. This means non-sister chromatids exchange DNA. Points where this takes place are called chiasmata. Next is metaphase I, during which homologous chromosomes line up on the metaphase plate. Kinetochore microtubules attach to both chromatids of each homolog. Next is anaphase I. During this phase, homologs separate and move towards opposite poles. Next is telophase. Cytokinesis creates 2 cells, each with a haploid set of chromosomes. Each chromosome is made of 2 sister chromatids. In some species, the nuclear envelope reforms and the chromosomes decondense. Next is prophase II, during which a spindle forms in each cell. After prophase II is metaphase II. Chromosomes line up on the metaphase plate, and the kinetochores of sister chromatids are attached to the microtubules of the spindle. Next is anaphase II, during which sister chromatids separate and move towards opposite poles. Last is telophase II. The nucleoli reappear, the nuclear envelope forms, and the chromosomes become less condensed. At the end of meiosis II, the cells undergo cytokinesis, during which the cytoplasm divides. The 4 resulting daughter cells are genetically distinct. They are haploid and have half the number of chromosomes as the parent cell.
For both mitosis and meiosis, DNA replication occurs in interphase before the cell cycle begins. Mitosis goes through prophase, metaphase, anaphase, and telophase once, and thus has 1 division. Meiosis goes through prophase, metaphase, anaphase, and telophase twice, and thus has 2 divisions. Synapses of homologous chromosomes and crossing over do not occur in mitosis, but does occur in meiosis. In mitosis, there are 2 daughter cells, each containing the same amount of chromosomes as the parent cell. Each daughter cell is genetically identical to the parent and it's diploid. In meiosis, there are 4 daughter cells, each containing half the amount of chromosomes as the parent cell. Each daughter cell is genetically different and is haploid. Mitosis is used for reproduction of somatic cells, single-celled organisms, and cells of a developing embryo. Meiosis is responsible for the production of gametes.